Somewhere in your body right now, there is a chemical mark that isn't yours. It didn't come from your genes being rewritten. It came from your great-grandparents' worst experience. If they lived through a famine, or a war, or a specific kind of terror, their bodies didn't just remember it. Their cells attached tiny chemical tags to their DNA in response, tags that changed which genes got turned on or off. Those tags weren't supposed to survive being passed down. But in some cases, they did. You inherited more than eye color and bone structure from the people who came before you. You may be carrying a molecular echo of an experience you were never present for. This isn't a metaphor for generational trauma. It is a specific, measurable, biological mechanism. Scientists have a name for it: epigenetics, the study of how experience marks the genome without rewriting it. It has been tested in real families, across real famines and real wars. The evidence is not simple, and it is not always comfortable. By the end of this video, you'll understand exactly what your body may have inherited, and why. To understand how this works, start with a basic distinction. Your genome is the sequence of DNA letters you inherited, fixed at conception, identical in nearly every cell in your body. Your epigenome is different. It's a layer of chemical instructions sitting on top of that sequence, telling genes when to switch on or off. The word epigenetics was coined in 1942 by British biologist Conrad Waddington. He imagined development as a ball rolling down a landscape of branching valleys, shaped by both genes and environment. Decades later, molecular biologists found the actual physical mechanism behind Waddington's metaphor. One major mechanism is called DNA methylation. Small chemical groups, called methyl groups, attach directly onto the DNA strand. They typically attach at specific locations called CpG sites, and their presence usually silences the nearby gene. A second mechanism involves histones, the spool-like proteins that DNA wraps tightly around inside the nucleus. Chemical tags on histones can loosen or tighten that wrapping, making genes easier or harder for the cell to read. Neither mechanism changes a single letter of your DNA sequence. Your genetic code stays exactly the same. What changes is which parts of it get used. Think of it as the difference between the words in a book and which pages have been dog-eared, underlined, or taped shut. The words never change. But what actually gets read, and how often, absolutely does. And unlike your DNA sequence, these chemical tags can be added or removed within a single lifetime, in response to real events. The clearest human evidence for this comes from one of the darkest winters in modern European history. In the autumn of 1944, Nazi forces blockaded food supplies into the western Netherlands in retaliation for a railway strike. What followed became known as the Dutch Hunger Winter, and it lasted until liberation in May of 1945. Daily rations in some cities fell below 900 calories, then below 600, forcing people to eat tulip bulbs and sugar beets to survive. An estimated 20,000 people starved to death before the famine ended. Because the famine had a precise, well-documented start and end date, it created something unusual: a natural experiment on human biology. Researchers could identify exactly which pregnancies overlapped with starvation, and exactly which trimester was affected. Decades later, scientists began tracking down people who had been in the womb during those months. In 2008, a team led by Bastiaan Heijmans at Leiden University Medical Center published a landmark study in the journal PNAS. They examined a specific gene called IGF-2, which regulates growth before birth. In people who had been conceived during the famine's peak, IGF-2 carried significantly less DNA methylation than in their own unexposed siblings. Less methylation meant the gene was more active than it should have been. This wasn't measured shortly after the war. It was measured 60 years later, in adults in their 60s. The chemical mark from a famine that ended in 1945 was still legible in blood samples drawn in the early 2000s. It is one of the longest documented persistent epigenetic changes ever recorded in a living human population. People exposed prenatally to the famine also showed, decades later, higher rates of obesity, diabetes, and cardiovascular disease. Their bodies had been chemically primed, before birth, for a world of scarcity that no longer existed by the time they grew up. It was a mismatch between the environment their epigenome prepared them for and the one they actually lived in. 60 years is not the outer limit either. Some of this research has now followed the grandchildren of those pregnancies. Grandchildren of women who were pregnant during the famine show elevated health risks too, despite never experiencing hunger themselves. This finding didn't stop with one gene. Later studies, led by researcher Elmar Toby and colleagues, expanded the search across the entire epigenome. They found altered methylation at genes involved in growth, metabolism, and even brain development, all linked to prenatal famine exposure. Timing mattered enormously. The strongest, most consistent effects appeared in people exposed during the earliest weeks after conception. Exposure just a few months later, in the same starving population, produced weaker or different effects entirely. This suggested the epigenome is most vulnerable during a narrow, critical window, right at the beginning of development. A few weeks of extreme deprivation, at exactly the wrong moment, left a mark that outlasted the person's own memory of it. Nobody exposed prenatally to the famine consciously remembers it. They were not yet born. Their bodies apparently remembered anyway, in a language written in methyl groups instead of memories. Human famine data raises an obvious question. Can this kind of mark be passed down to children who were never exposed at all? Animal research has tried to answer that question. With tightly controlled experiments, no one could ethically run on people. In the early 2000s, neuroscientist Michael Meaney at McGill University studied how mother rats care for their pups. Some mother rats licked and groomed their pups constantly. Others did so rarely. Meaney's team found that pups raised by high-licking mothers had different methylation on a stress hormone receptor gene in their brains. Those pups grew into calmer adults, with a milder physical stress response for the rest of their lives. Pups raised by low-licking mothers developed the opposite pattern, heightened, long-lasting stress reactivity. Crucially, this had nothing to do with the pup's own genetics. When researchers swapped pups between high-licking and low-licking mothers at birth, the behavioral pattern followed the caregiving, not the biological parentage. A calm biological mother's pup, raised by an anxious foster mother, grew up anxious. The chemical mark was installed by early experience, not by inherited DNA sequence. It showed that a mother's behavior could directly rewrite a specific gene's activity in her offspring's brain. It also showed that these marks, at least in principle, could persist and shape an entire adult life. A more startling demonstration came in 2014 from Dias and Kerry Ressler, then at Emory University. They trained male mice to fear a specific smell, the scent of cherry blossoms, by pairing it with a mild electric shock. The mice, understandably, learned to freeze and show fear at that smell alone. Then the researchers let those mice father offspring, without ever exposing the offspring to the smell or the shock. The pups, and even the grandpups, showed heightened sensitivity to that exact same cherry blossom scent. None of them had ever smelled it before, and none of them had ever been shocked. Dias and Ressler found structural differences in the offspring's odor-detecting neurons, tuned toward that specific smell. They also found altered methylation on the gene responsible for detecting that odor, passed down through sperm. Publishing in the journal Nature Neuroscience, they described it as the transgenerational inheritance of a specific learned fear. A single frightening experience, in one generation, appeared to shape the nervous system of generations that never lived through it. The study remains one of the most cited and most debated papers in the entire field. Critics have questioned exactly how a memory this specific could survive the reset that happens between generations. But the core finding, that a father's specific experience altered his offspring's biology, has been difficult to fully explain away. Human data pointing towards something similar comes from a very different, very human source: the children of Holocaust survivors. Psychiatrist Rachel Yehuda at Mount Sinai in New York has studied these families for over 2 decades. Her team examined FKBP5, a gene central to regulating the body's stress hormone system. Children of Holocaust survivors showed methylation patterns on that gene, distinct from demographically matched Jewish families without direct trauma exposure. These children also showed altered cortisol regulation, the hormone most directly tied to the body's stress response. Many of them had never experienced anything close to what their parents survived, yet a measurable biological signature of that trauma appeared to be present in their own bodies. Yehuda published these findings in 2016 in the journal Biological Psychiatry, drawing international attention. She was careful to note that human data like this cannot rule out every alternative explanation. Upbringing, culture, and secondhand storytelling can also shape a child's stress physiology, without any direct chemical inheritance at all. Even so, the biological signature was specific enough to demand a serious scientific explanation. Here's the part most retellings of this story leave out. During the formation of sperm and eggs, and again in early embryos, the body runs an aggressive reset process. Most epigenetic marks get systematically erased and rebuilt from scratch at that stage, specifically to prevent this kind of inheritance. This erasure evolved for a reason. A parent's temporary environmental damage usually shouldn't become a child's permanent inheritance. That means transgenerational epigenetic inheritance, if it happens in humans, has to somehow slip past the body's own safeguards against it. Exactly how certain marks might escape that reset is still an active, unresolved area of research. Some scientists remain genuinely skeptical that clean transgenerational inheritance, as seen in Dias and Ressler's mice, fully applies to humans. What almost everyone agrees on is narrower, but still remarkable. Prenatal experience alone can leave decades-long chemical marks, as the Dutch hunger winter proved beyond serious doubt. Whether those marks can jump cleanly across an entire generation untouched remains one of the most contested questions in the field. Think about the people who came before you, the ones you never met. Somewhere up your family tree, someone survived something that should have been unsurvivable. A famine, a war, a loss so total it reorganized how their body prepared for the future. You may be carrying a small, specific trace of that moment, written not in a story, but in chemistry. Not a metaphorical burden, an actual methyl group, sitting on an actual gene, inherited the same way scar tissue outlives an injury. This isn't fate, and it isn't a life sentence. Epigenetic marks are, by definition, more flexible than your DNA sequence ever could be. Diet, stress, exercise, and environment can all shift methylation patterns throughout your own lifetime, the same way your ancestors' experiences once shifted theirs. The chemistry that arrived with you is not necessarily the chemistry you'll pass on. That is, in its own way, a strange kind of hope hidden inside a heavy discovery. You are not simply a fixed inheritance, arriving fully sealed from people who lived and died before photography, before antibiotics, before you. You are an ongoing negotiation between an ancient genetic code and everything that has happened to the people who carried it. Somewhere in that negotiation is a famine survivor's malnourished body, quietly instructing a great-grandchild's metabolism it will never meet. Somewhere in it is a mother rat's grooming, echoing forward into a calmer, or more anxious, adult decades later. Somewhere in it, possibly, is a fear nobody alive today experienced firsthand, still humming faintly in the body that inherited it. None of this makes you less yourself. If anything, it means the self you experience as fixed and singular is actually a living record, still being written. Every gene you carry is real, permanent, unchangeable text. Every mark sitting on top of it is closer to an annotation, added by lives you will never fully know. Some of those annotations may soften with time. Others, like the Dutch hunger winter's IGF-2 signature, may last 60 years or more. Somewhere inside every cell in your body, ink from a hand you never shook is still faintly legible. That ink is not your whole story. But it is, unmistakably, part of the sentence.